Okay, thank you. There's a really neat study that was published a couple of months ago by Maria Dornalis, who was actually a postdoc in the COE in Program 2, and she's moved on. But what she's done, her and her colleagues, was she mapped took a number of data sets, global data sets, long time series, and looked at the alpha diversity since 1900 to the present, and also beta diversity. And they found that alpha diversity was essentially constant through time. In other words, no systematic species loss across the globe, whereas beta diversity always showed a downward trend. So this is simply that communities are becoming less and less similar through time. One of the primary reasons for this disparity in these two graphs is the migration of new species and species invaders into new areas, many of which become into areas more suitable. They become more suitable for habitation due to climate change. And I think this is really a call for critically examining patterns of species movement in marine systems. Climate change challenges organisms to adapt or move to track changes in environments in space and time. And one can make predictions about where those movements are going to occur by looking at temporal changes in sea surface temperatures, well, temperatures on the land and in the sea. And this is the so-called velocity of climate change. And one could map these areas of high change and low change through time. So the expectation is for a dramatic change in taxonomic composition of ecological communities as marine species respond to warming by shifting their distributions. So there's some nice examples of this already in the Northern Hemisphere. This is a compilation done by Pinsky and co-workers that showed that marine species shift at different rates and directions, but basically in line with the predictions from a velocity of climate change situation. In other words, they're tracking the complex of mosaics of local climate velocities. Well, another study has shown that this is actually happening not just in the North Atlantic, but all over the world. So this paper by Poleward et al. Raised the number of known marine impacts to climate change from 85 in the 2008 IPCC reports to 1,735, which will be coming out in the new IPCC reports. But essentially, distributional changes and lots of other changes are occurring over a broad range of marine taxa. Well, what about the ecosystem of the hour, our beloved coral reefs? We've had lots of responses to global warming through migration. There's increased documentation of these migrations in Japan, where a number of species groups of Acropora have been marching northwards in Japan over the last 80 years to the tune of about 14 kilometers per year. In Florida, we have documentation now of Acropora cervicornis thickets, which had been confined to this area, but now are moving northward in conjunction, well, in the same places where they used to live in the Holocene. This didn't come out very well, but in 1988, a living species of Acropora was also found for the first time in Rottnest Island. So there's evidence for migrations on the west coast of Australia, and then also on the east coast of Australia. We've got several species now in the solitary islands that we didn't have before. Well, so today I just wanted to focus some attention on what I'm calling the tip and the tail of coral species distributions, by using an example from two vignettes, one from the subtropical Eastern Australia, the leading edge of coral distributions in Australia, and also another study looking at the tail of species distributions by comparing global diversity gradients between modern and Pleistocene occurrences. And then I'll end the talk with some reflections and recommendations about what this sort of information might mean for the future of coral reefs. So going to the tip of the Eastern Australian coral distributions, one of my PhD students, Bridget Summer, has conducted this study. So if you want to know more about it, or if I make any mistakes following Hugh's lead, then you can go and see Bridget. But what she was looking at is the species composition of coral communities along a large tract of Eastern Australia, on the southern subtropical communities. We're going to look at diversity gradients, zoogeographic composition, and very briefly at species traits, the distribution of species traits. The study area runs from the Sunshine Coast in the north down to Port Stephens in the south, about 6 degrees of latitude, about 17 locations. The leeward side of the islands and the headlands were targeted to keep habitats as constant as possible for comparisons. The data collection involved replicate 1 meter phototransects in about 8 to 12 meter water depth. Bridget then used the CPCE method to identify the corals and make abundance accounts of the relative abundance of the corals along her transects. Now, one of the things that we did was to divide the corals into different zoogeographic categories, mainly from Charlie Viren's monographs about distributions of these species, not only in Australia but worldwide. The tropical species were the ones mainly occurring in the tropics. The subtropical species were those that were rare in the tropics but common at high latitudes, including some endemic species. And the cosmopolitan taxa were those that were common throughout their geographic distribution. Now, what happens? Well, first of all, something quite unsurprising. The latitudinal diversity gradient that's been known for a long time on the eastern coast of Australia is quite pronounced in the data set. As you move from lower latitudes to higher latitudes, we get a drop in both species richness and in Shannon-Wiener diversity indices. But what about the distribution of these zoographic distribution of these taxa? This is a plot of the number of sites that a species occupied versus the number of species that occupy those sites. And you can see that most of the species, about 60% of them, occur in only 1 or 2 of the 17 sites sampled. The majority of the species then had very narrow distributions, and only about 11% of the species occurred at 10 sites or more. The cosmopolitan and the subtropical species, the blue and the green, were distributed a bit more widely than the tropical species. Now, the thing to notice here is that the vast majority of species that we ran into were tropical species, 76 out of only 9 subtropical and 7 cosmopolitan. However, when you look at the relative abundance, you find the story is quite different, that throughout most of the study sites, the cosmopolitan taxa, only those 7 taxa, have most of the abundance on these reefs. And you find that the tropical species, while they're quite abundant in the more northerly localities, the warmer localities, quickly drops off in their abundance as we proceed further south. So the relative abundance of the tropical species is high at the northern site and then diminishes with increasing latitude. Well, I like to sort of picture these things in ordinations. They help me visualize, and they help Bridget visualize what the factors are that are driving those distributions. And this is a canonical analysis of principal coordinates, and it shows the distribution of how similar these communities are to one another. And the subtropical communities obviously are more abundant in the south, with the tropical species occurring more abundantly in the north, and also farther offshore. So this is an offshore to onshore gradient here. So basically, latitude and distance from shore explain about 95% of the variability in community structure. The tropical influence is both towards the north and offshore. And the subtropical endemics are the highest, contribute the most at the highest latitudes. So very briefly, we also gathered a whole lot of information on species traits, morphological traits, reproductive traits. We even used Emily Darling's life history classification that she based on the Grime classification for plants. And what we found is if you plot species co-occurrence with the functional distance, that is the distribution of traits among those species, you find that the co-occurrence drops off as the functional distance increases. So things that share more and more species traits also tend to co-occur together more often. So I'll get back to that result as when I get to the other vignette. So just a question before I do that, and that is, can high-latitude coral communities host abundant and self-sustaining populations of the tropical coral species? Maybe, but the capacity is probably limited for a number of reasons based on these data. There's low abundances of the tropical species at most of those high-latitude locations. We're talking about fragmented habitats. Most of the east coast of Australia is characterized by moving and shifting sands, so there isn't an opportunity to develop a continuous sort of reef structure. And any of those new sort of migrations are going to have to depend on tropical source populations much further north. When you couple that with things like acidification and human impacts, then you really must be quite measured if you're thinking about what the capacity is for these communities to march down the eastern Australian coast. Okay, and I'll get back to that as well at the end of the talk. But now let's go to the tail of equatorial distributions, that is looking at the trailing edge. And for this, I'm going to call upon a couple of global databases in comparing modern species distributions with species distributions in the late Pleistocene. The time that I'm going to focus on is the last interglacial marine isotopic stage 5E, which was warmer than present day. So basically, we're looking at a comparison in the past, using the past as warm and today as cold. So that's probably not quite the way you normally think about things, but let's think about today as cold and the Pleistocene as warm. So we looked at we did a global comparison of the distribution of coral communities. This is a paper we published at the end of 2012 with Wolfgang Kiesling with this Pleistocene and modern context. So we gathered all the data we could on modern reefs, shown in orange here. This is sort of spatially explicit geographic occurrence of species occurrences. And the fossil reefs are in red, obviously a whole lot less coverage for the fossil reefs. But so what you do in that case is you degrade the modern data set so that you can make the modern and the fossil data sets comparable. Even the data sets we used, we present as preliminary because we're enhancing these data sets as we speak, and I can talk to you more about that after the talk if you like. These are so-called beam plots. They provide densities of species leading edge and trailing edge range limits. In the northern hemisphere, the red is the last interglacial, remember warm. The orange is recent, cool. And you can see relative to the modern, the warmer Pleistocene has more poleward extension of range extensions for the leading edge, and the trailing edge also shows a retraction away from the equator, okay? And the same thing happens in the southern hemisphere with a poleward displacement in the warmer Pleistocene period, and also displacement away from the equatorial tropics for the trailing edge populations relative to the cooler modern periods. So we've got these differences in species ranges. Now, what we can do is we can draw species diversity maps with latitude. And with the data that we had, this is what the curve looks like for the modern, with a pronounced equatorial, well, I'd say more subtle equatorial dip here for the species diversity. But let's look at the Pleistocene. In the Pleistocene, this equatorial drop was quite a bit more pronounced, about 35% off of the northern hemisphere higher diversity areas and higher latitude areas. So we've got a substantial loss or decrease in diversity associated with this warmer time period. Now, speaking about traits and what you can do with this kind of data, obviously, different species are going to have different distributions. Some are going to be distributed in both the northern and the southern hemisphere. Some of them are going to be in the north and the south, but not equatorially distributed. Some no equatorial distribution, but only in the north. Some no equatorial distribution and only in the south. So you can take information like this, gather information on the traits, and begin to start looking at what are the drivers in life history characteristics that are making these species more and less vulnerable to life either in the hot zone of equatorial tropics or at the edge zone in the leading edge. And you can do the same thing with the fossils. So our last two slides, I'll just sum up what's this information tell us. Well, when we think about poleward increases in reef development, we also have to think about the offsets that are going to occur by way of equatorial contractions. So life isn't just what's going to happen as the reefs march further south in the southern hemisphere or north and northern hemisphere. It's also about what's happening in those equatorial regions. Coral species diversity drops off from both the trailing edge and the leading edge of their distributions. So we've got things happening in both areas. Species loss is likely to be non-random at the range margins, so we can have this expectation of altered reef communities. Going back to Dornalis's study, these are the kinds of things that we can expect. Lots of turnover, maybe not so much species loss, but lots of changes in the sorts of communities that we're going to find. And I think that coral species traits are going to influence the vulnerability to extinction at these range margins. And I think this is a really fruitful future area of research to try and go and look at the distribution of traits among these taxa. Because face it, it's probably a bit easier to get those traits than it is sometimes to do the coral taxonomy. My last slide, just a few recommendations. I think there's a clear need to understand the ecological dynamics of tropical, equatorial coral communities. Life extends beyond Heron Island and Lizard Island. We should be doing a lot of work in these equatorial regions, maybe more than what we normally think about doing. Also, a call for focused and dynamic management strategy that takes into account future changes at the species margins. Think about these margins in a dynamic sense, how they're going to change, and look at it not only from a taxonomic viewpoint, but also from this idea of life history traits and other kinds of species traits. And lastly, I think there's important information that needs gathering on the past response of reef species, and not just during the extinction events or the PETM, or there's a lot of information just in the normal sort of day-to-day or million-year to million-year goings on of reefs through time, and we need to focus in on the parameters and the relationship between the environmental parameters and the biotic response on a more subtle level, I think, than perhaps we tend to focus on. Thank you very much.